Okay, hi guys, welcome to Biology Online with Henny van Vieren. Hello again guys, welcome back. I hope you're surviving and you're not getting cabin fever and everybody is staying healthy. Um, it looks like this is kind of the new normal, so we will see how these things go. Anyways, today we're going to talk a little bit about active transport. Yesterday we covered passive transport and specifically osmosis. Now you need to remember that osmosis is the movement of specifically water from an area of a high water potential or a low solvent concentration to an area of low water potential or an area of high solvent concentration across a semi-permeable membrane. So the point is, water always wants to dilute the concentrated stuff until the concentration of both sides of the membrane is the same. So water wants to dilute things down, and that is osmosis. So today, as I said yesterday, we looked at passive transport, and passive transport does not require any ATP. But today we're going to look at active transport, and as mentioned earlier, we for this it actually does need ATP. Okay, so on with the lesson. Great. So passive transport, we said, is diffusion, osmosis, and facilitated diffusion. Remember, facilitated diffusion is when there's a, um, a protein involved, a membrane protein or membrane-bound protein, something specifically like an aquaporin or a channel protein, where stuff that are water soluble can go through, whereas stuff that is fat soluble can simply travel through the membrane. And the other three little molecules that can travel through the membrane without having to go through a protein is oxygen, carbon dioxide, and water. So passive transport is always along the concentration gradient and no energy is required. So here we have active versus passive transport. When you look at passive transport, it is simple diffusion. This could be oxygen, carbon dioxide, or water just traveling through the membrane, and don't forget that it can also be fat soluble molecules. Then we have a channel protein. This is facilitated diffusion. Both of them use proteins. Channel will simply allow water soluble stuff to come through, whereas a receptor protein will receive it, and because it's binding, it actually changes the changes the actual shape of the protein to release the molecule on the inside, and then the shape changes back. Now with active transport, we need ATP, okay, adenosine triphosphate. It's going to give off one of those phosphates that will give energy, and the energy will make this thing change. The reason why we have that is that we can sometimes have a very low concentration gradient, so diffusion is going to happen really slowly, so we might have to speed that up, or we can have a moving of molecules against the concentration gradient, so building up a bigger concentration gradient, or reuptaking nutrients and so forth. So you can think about in your intestines, further you go along, the lower the nutrient content, and we have to actively transport that into the cells. Okay, so the movement of molecules or ions through a transport protein across a cell membrane against their concentration gradient using energy from ATP. It's when diffusion is not possible and it always requires energy. Okay, let's look at some examples. Plants need to take up minerals and salts from the soil. But how do you take minerals and salt out of an issue and into your cell itself? Because the cell's vacuole is basically filled with water. So the hair root has to actually actively pump these salts and stuff in to bring it into an area of a already higher concentration than the outside. So consequently it is going against the concentration gradient, because in the water itself, in the soil, the concentration of these mineral salts are very low. So in comparison to the cell's vacuole, there's less dissolved stuff in the soil water. So the cell has to actively pump mineral salts and so forth into the vacuole. The water will move passively across to try and dilute down or create equilibrium with the cell's vacuole. Now, when cells want to create a large concentration gradient across the membrane, such as in the kidneys, this happens a lot. In the kidneys, we need to reuptake glucose and so forth, and it is very important that we can get those across the membrane. And then also when diffusion is not fast enough, like when we have really big molecules or ions or molecules will be actively transported um, uh, with the concentration gradient to make the whole process go faster. Okay, so if you look at this little image here at the bottom, what's happening is this is a sodium potassium pump. So we have sodium, and then we have, sodium is yellow, potassium is green. And you can see currently we kind of have equilibrium, but as the potassium is pumped out, it is exchanged for the sodium, and you can see how we're building up this concentration gradient for both of them. But it requires energy every time, and that's where the ATP comes in. ATP gives the energy, it actively pumps it against the concentration gradient. Actively, when the phosphate is released, it actively pumps it against, and you can see the phosphate has opened it. Your potassium goes in, and as it releases, it will go back to its original state. Okay, so integral protein pumps are very, very important in our bodies. They are usually very specific, and they get their energy from ATP. So when I don't believe there's audio in this. So this is the same thing, the sodium potassium exchange pump. So you can see for the sodium to come in, we have this transmembrane protein or a transport protein. We will have sodium going in, and then it needs ATP. The ATP binding site is now active because the sodium has gone in. ATP will come give off one of its phosphates. As it releases, the energy is released, and this changes the shape of the molecule, and then the sodium can be released towards the outside of the cell. When potassium comes back in, now potassium can come back and bind on the potassium side of things. When that happens, the shape of the protein will change to release this phosphate, and as it releases the phosphate, the whole thing changes its shape again. And now it is open for the sodium to be able to go out again. Okay, I hope that makes sense. Okay, so here's another one where we have active transport, and this is in the kidney. Okay, and co-transport or coupled transport, the concentration gradient of one molecule or ion drives the active transport of another. So in other words, if you can look here, there's a lot of water on that side. And in the blood, you can see this is blood because we have the red blood cells over there. We have a few more molecules currently of sodium and glucose. Okay, I believe they labeled them wrong because if you look at it, glucose is supposed to be a hexagon, but never mind. You will get the idea. So if you create suddenly a lot of dissolved solute on that side, water will follow. Or if you do the same on this side, water will follow. So have a look at this. This is the sodium glucose pump. So when you have a lot of dissolved stuff here, you pump sodium and glucose through, and because this concentration is now so much higher on this side, water will go through passively. Let's look at that again. So we're artificially creating a concentration gradient to draw water out of the bladder back, or out of the kidney and back into the blood, not to lose too much water from urine or in urine. So we pump them actively using ATP, and then creating this concentration gradient, the water will follow. Pumping them actively, suddenly we have a big concentration gradient here, not as much on that side, and the water follows. Okay, so active transport. Now we also have something called bulk transport. Just like when bulk shopping and everybody's who panic bought and bulk shopped lots of stuff. Bulk transport is when we're not only taking in little molecules but a lot of molecules at a time. So one of them is endocytosis. Okay, endocytosis. It's the bulk movement of liquids. Liquids is when we cell drinking, pinocytosis, or solids phagocytosis into the cell by the infolding of the cell's surface membrane to form vesicles containing the substance. Endocytosis is an active process that requires energy from the ATP. Now you will remember that amoeba that consumed the two the two paramecium that I showed you the video. It's later on in this too. They have to use their pseudopods. They went around the prey and then engulfed them by phagocytosis. You will see that again very soon. Okay, phagocytosis is cell eating done by phagocytes like your white blood cells. Pinocytosis, cell drinking. Okay, you also get micropinocytosis, so like the human egg cell taking up nutrients. Then we have exocytosis. This is the exo, in other words, sending it through the exit. It's the reverse of endocytosis, and it is to excrete. It happens in the pancreas with the secretion of digestive enzymes and so forth. Okay, so here you can see this would be, again, I don't know if there's there seems like there's audio in here.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system.
This is what
the phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
Okay, I think that explained it nicely. So we call that bulk transport. Bulk transport, exocytosis, endocytosis in the form of phagocytosis. Okay, this was the video of the
neutrophils are white blood cells
neutrophil
that hunt
as it
and
chases
kill bacteria.
Staphylococcus aureus, and you can see phagocytosis
And engulfs them
happens
by
basically
phagocytosis.
there. Now it's inside and the lysosomes will take over. This one is where, and again, if you want to look at these videos, they were in the presentation. You can see there's pseudopods going around these two paramecium, and as it engulfs them, the lysosomes will come. Lysosomes are going to be released. With those phagocytes form, so bang, it's going to be released now, and they're starting to be digested. Eventually, after taking up all the nutrients it needs, it can have the waste products just go through exocytosis. That is the end of that. So, active transport, guys, in a nutshell. I hope you guys followed that, and I will put some work on to Education Perfect for you. Thanks a lot. Have a great day. Bye-bye.